Hey everyone, my name's William Polk. I'm here in this genetics class, and I want to go ahead and get started with the lesson 1 group problems. So on question number 1, it says that you're going to perform a cross between big A, big B, big C, big D, big E, and then to an big A, big B, big C, big D, and two little e's. And because if you guys can read my notes, since they're very easy to read, you'll be able to just see that I counted them all out. I FOILed them, did the proportions for each one, and how it would be. You just go ahead and multiply them together. So question number part A says, what proportion of the offspring is expected to have the genotype that is listed? And I got 1 over 128. For the subset B, a similar question, it was 1 over 256. The next one, let me see. So part C was actually kind of a trick question because it asked about what percentage will come out with 2, which is the recessive of the D alleles, and there's actually not, because there's a cross between 2 of the dominant ones and a heterozygous of the big D and the little d. It actually has 0 of them that are going to come out there with that genotype of little d's. The next one is subset D. It says what proportion of the offspring, and it's 1 over 32. So for part 2, it's actually just a fill in the blank section for vocabulary terms. And the first one, it says paired homologous chromosomes are sometimes called bivalents or tetrads. Letter B says the structures that connect centromeres to the microtubules of the spindle. It's called a kinetochore. Part C is talking about a fruit fly, and in prophase 1 it has 16 sister chromatids, while at the end of metaphase 2 it actually only has 8 sister chromatids. So it's a dihybrid cross. This is the type of cross that you do to be able to see if the purple flower plants have the big P little p or the big P little p genotype. Because of those criteria, you can actually look at the phenotype and see which ones are going to come out. Subset E says a non-functional allele is called an amorphic allele, and the one that is partial function allele is called a hypermorphic allele. Subset F is actually about the 2 different laws of Mendel's laws. The first one is the independent assortment, which involves 2 different traits, while the law of segregation actually only involves a single trait. Alright. So number 3, it is actually a little bit harder to explain because you actually have to see a lot of work. But in subset A, it says for you to draw different ones. I have 4 different sets of ones that it can be, and it indicates which alleles are found on which ones. And then it says B, which is indicate all the possible gamete types. So I have 4 different ones. I have big T, big R, big Y. The next one is big T, big R, little y. The next one is big T, little r, big Y. And the last one is big T, little r, little y. And so it says how many sister chromatids are found per cell in the metaphase 1. And the answer that I came up with is there's 12 sister chromatids in 1. And how many chromosomes are found per cell in the organism during metaphase 2. And I said there's 3. The next question is 4. It's about a horse that has 32 pairs of chromosomes. The way I came up with this one is if it has 32 pairs, there's actually 64 chromosomes because they're in their pairs of 2. So the first one is how many chromosomes are present per horse cell at the conclusion of meiosis 1. And it's actually going to be the same because it's just a duplication for meiosis 1, which would be 64 chromosomes or 32 pairs. The B set is how many tetrads are present per horse cell during prophase 1 of meiosis, and there are 32. How many sister chromatids are present in a horse cell during prophase 1 of meiosis, and I found 128 because there are the 64 different chromosomes. The sister chromosomes are actually double that. Next one, it says how many sister chromatids are present per horse cell during metaphase? 2, which is 64. And then the last one is how many bivalents are present per horse cell during anaphase of meiosis? And I said that there are 0 because they should all be separate at that point. They're no longer going to be in those pairs. Alright, so let's get to the last question. The last question is 5, and it's talking about the different proportions of 2 different crosses. In the different crosses I have, what proportion of the offspring would be tall, round, green, and terminal? I have that there was 1/64 of one and 1/16 of the other, so when I add them all together, you make the common denominators the same, and it was 5/64 of a is your proportion of what it would be. Because I think of it like this, they're asking for the dominant trait, so it can be expressed as a homozygous or also a heterozygous, either or, and they would both show those phenotypes. What proportion of offspring plant has the little t, big R, big Y, big A, little a genotype? And after I pulled it up into all the fractions and multiplied them all together, it came to about 1/32. What proportion of offspring would be heterozygous for all 4 genes? Would be 1/16 of all the offsprings that will be heterozygous for there. And it says what proportion of offspring will be dwarf, wrinkled, yellow, and axial? And I have 1/64 of all the offspring will show those genes. And if you'd like to be able to see my work and the different things, I have uploaded a picture. I can always, if you just request, I will upload a picture of my work to be able to show it a little bit better on how I counted it up. But most of it is pretty simple. Most of it was explained on the PowerPoint presentations, the vocabulary terms, and also there was a video on how to do the different kind of crosses. And after watching the video, you could be able to count up the proportions, just multiply them all together on the different options, and that would give you your answers. And that's going to be all for this week, and you guys have a great rest of your week.